My name is Brian Smith, and I'm in the laboratory of Bradley D. Smith at the University of Notre Dame. And today I'm going to be talking about optical imaging of anionic membranes for detecting and monitoring disease states in vivo. So in biomedical research, there are two main groups, those that do anatomical imaging and those that do molecular biology. Now, anatomical imaging uses the inherent properties of tissues to measure bulk anatomy. Now, malignant tissue and normal tissue will have different contrasts. So we can see this brain tumor can be delineated from this normal brain tissue. However, this not tells you the underlying biochemistry of this cancer. That's where molecular biology comes into play. Molecular biology allows you to study single pathways that are very important for disease processes. Well, recently, these two fields have come together to form molecular imaging, where you can then study the physiology and biochemistry of a malignant tissue in its native environment. However, in order to study this biochemistry of the tissue, you need some kind of reporter, whether it's a genetic fluorescent reporter or exogenous molecular probe. So this is a common architecture of molecular imaging probe. We have our affinity group in the blue. This can be a peptide or antibody. It's basically something that's just going to target your molecule of interest. And then this is linked to a reporter group. And this is going to change depending on the modality that you chose to use for imaging, whether it's a radioisotope or, in our case, a fluorophore. There are also many other types of molecular probes. A well-used molecular probe now is the activatable probe, and it is a probe that has a fluorophore and a quencher. Both of them are linked with a group in the middle that can be cleaved by an enzyme. So these are used to study disease processes where certain enzymes are overexpressed. You also have multivalent nanoparticles and dendrimers that just use this idea of multivalency to target molecule of interest. Whether you do an microscopic or macroscopic fluorescence imaging, you have a wide variety of fluorophores to choose throughout the light spectrum. Many common ones are coumarin, BISD, Di3. However, for in vivo imaging, we're going to focus on this near-infrared region, which is between 650 and 1,000 nanometers. And these include fluorophores such as Di5, Di7, and ICG. Now, we want to focus on these fluorophores because, one, in the near-infrared region, we have less, in the blue-green filter set, we have a lot of autofluorescence coming from the intestines, the bladder, and also the skin. The same is true with the green-red. You see this autofluorescence in the intestines and the skin. However, when we get to the near-infrared, this autofluorescence has been severely attenuated. Also, there's high sensitivity. In this image here, we're looking at protease expression levels, and this is without a drug. However, when you do administer a drug, you can see that the protease expression levels have been severely decreased. It's also safe and non-invasive. You do not use radiation. Therefore, you can use one animal for many time points, which will give you more statistically relevant data. It's extremely cheap, and you have high resolution, and you can see that we can image tumor foci in the wrist of a mouse. So all my images have image stations, with most of them coming from the multispectral In-Vivo FX. So this is just a cartoon of how the multispectral works. So we have a mouse, and it has some kind of malignant disease, and it was injected with a fluorophore. We place it in an image station, and when we acquire the image, we'll accept the fluorophore with a certain region of light, and then it will emit at a longer wavelength. The camera will then pick up this wavelength and relay it into an image such as we see here. Now in this image, we see different gradients of fluorescence intensity, the white being the highest. And with most of the fluorescence intensity localized to this region in the shoulder, which in this case just happens to be a tumor. So my talk today is going to focus on anionic cell membranes. Just to give you a quick background bilayer. And on the outer leaflet of the bilayer, we have a near-neutral charge due to phospholipids such as phospholipid and also abundance of cholesterol and glycolipids. However, in the inner leaflet, we have anionic phospholipids such as phospholipid and phosphatidol and inositol. Gram-negative bacteria have a high abundance of anionic charges, and this is due to phosphatidylglycerol in the cell membrane, and also LPS, which is lipopolysaccharide. Also, gram-positive have anionic charges due to phosphatidylglycerol in the membrane and the acetylglycan layer. So there's a lot of biomedical importance for anionic membranes, and as we were just talking about with bacteria, so bacterial infection can be imaged and detected as anionic charges, and this is due to the flipping of phosphatidylserine from the inner leaflet to the outer leaflet. The same is true with tumor vasculature. It's been shown that the endothelial cells will expose phosphatidylserine, and viruses will expose phosphatidylserine in order to mimic apoptotic cells in order to enter living cells in the body. Now we are going to target anionic membranes through the use of this affinity ligand called a zinc-2-dipicolamine, or I'll refer to it as the zinc TPA. So the two dipicolamine groups will coordinate to zinc ions, and this will then allow for detection of both phosphorylated species and anionic charges. So we have conjugated these zinc TPA groups to a wide variety of fluorophores that range throughout the light spectrum. So this structure right here, the zinc TPA, has been conjugated to an anthracene, and we call this PSS380. This probe, the zinc TPAs, have been conjugated to a fluorescein. This is PSS480, and in this structure, the fluorophore is a carbocyan dye, and this is our near-infrared PSS794. So in order to actually show that we can target these anionic phospholipids, we made a series of vesicles and just did titrations. So here we have phosphatidylcholine, which is near-neutral phospholipid with phosphatidylserine, another anionic phosphatidylglycerol, and also another anionic phospholipid of phospholipidic acid. And with our control, just being 100% of the phosphatidylcholine. It's occurring with the phosphatidylserine vesicles next with the phosphatidylglycerol and the phospholipidic acid. And this is severely higher than the phosphatidylcholine, and this is just shown through the binding affinities. So our zinc TPA probes have the highest binding affinity for the phosphatidylserine, and this is greater than both phosphatidylglycerol and phospholipidic acid, and all three of these are significantly greater than the phosphatidylcholine. So now we can show we can target anionic phospholipids. We want to know if we can then target cell death. So we used our PSS480 probe to determine whether or not we could bind to cell death. So we treated turgid cells with camptothecin, which is just a cytotoxic agent, for 4 hours, and then we imaged under epifluorescence microscope that were stained with our PSS480. And if we look at panel B, these are cells that were stained with a nucleic acid dye 7-AD. Now this dye can only get into cells if the membrane's been permeabilized, which occurs during necrosis. So the most important image in these panels is C. So if we just look at the cells that are circled, these represent cells that are stained by our probe but not the 7-AD. So they represent cells that are undergoing apoptosis. But what's also interesting is that our probe can both detect apoptosis and necrosis in these cells. We also wanted to see whether or not we could image bacteria, at least in vitro. So we used both gram-negative and gram-positive bacteria. A gram-negative E. coli, we can see staining with the delineate better the staining of the membrane with hardly any staining in the center or in the inside of the cell. So this is showing that our probe is targeting these anionic charges in the gram-negative E. coli. And it's true about the Staph aureus. You can see good staining with the symptom and also on the membrane of Staph aureus. So now that we can target both cell death and bacteria in vitro, we want to see whether or not we can detect it in vivo. So in this experiment, we injected Staph aureus into the leg, and we let it incubate into the leg of a live mouse for 6 hours, and then we injected our near-infrared PSS-794. So this is a montage of images representing different time points post-injection of our probe. So this is 0, 3, 6, 12, 18, and 21 hours. So I just want to go through a couple things in these images. First of all, we can see that the probe diffuses throughout the body and that the whole body is lit up with high fluorescence intensities at least 0 hours post-injection. So over time, the body fluorescence significantly decreases, and this indicates that the probe is clearing away from the body. However, if we would look at the target site, the site of the Staph aureus injection, we can see that the fluorescence intensities increase and become very localized to this one area. So this is showing that our probe eventually diffuses through the body and then localizes to the site of the bacterial infection. And we can quantify the targeting ability of a probe through a target to non-target ratio of interest around your target site. And then you get the mean pixel intensity of the site. And you'll divide it by the non-target site, which is the same ROI that's on the opposite, in this case, in the opposite leg of the animal. However, it can be some other place on the body. For instance, you could have a non-target of the liver, but in this case, we just call that a target to liver ratio. So you take this target to non-target for each time point, and then you can graph it. So here is our target to non-target graph. And at 3 hours, we have the target to non-target ratio of 4, and this pretty much stays the same throughout. And our target to liver shows a different curve with our highest around 2.5 at 21 hours. So this is a good indication, just showing that at least 3 hours after infection. So after we sacrificed the animals after 21 hours, we did ex vivo imaging, and you usually do this in a in vivo study because you want to know the probe biodistribution if it's getting into other organs that you can't see in vivo. So we did both uninfected and infected mice, and then we draw an ROI around each organ and get the mean pixel intensity, and we can graph it as you see through right here. And the treated, we have a high accumulation of a probe in the left leg, which happens to be the leg of the bacterial infection. And this is significantly more than all the other tissues and the control. In which this case, the control is a controlled probe, which is just a fluorophore without the DTPA ligand. So both in vivo and ex vivo, we've clearly shown that we can detect bacteria, which is very important for biomedical cases. So next we wanted to test whether or not we could detect tumors. Now tumors, as I explained earlier, the vasculature will expose phospholipid. However, if tumors get large enough, they'll form this necrotic core due to the inability of nutrients to get to the center, to get to these center cells. So we used a mouse model that contained a memory xenograph on the shoulder, and we injected our near-infrared PSS-794 via the tail vein. This is an image that we received 24 hours after 794 injection. And you can see the high accumulation of this probe to the site of the tumor and how it is very localized to the site and not into other, to display an image that you can create, that you can acquire. Another way to show how the probe is localizing to the tumor is basically the montage, which I just showed through the bacterial infection. So, just like with the bacterial infection, the probe will diffuse throughout the body, and then that the thrust intensity will decrease by the end of the study, both in the body and in the tumor. But if we just look at this image, comparing the 24 to the 0, if we look at the site of the tumor, it appears that there's a lot more fluorescence intensity in the tumor at 0 hours than 24. This is because all these images are scaled to the same calibration bar. However, we just take this image to itself and use a different calibration bar. So instead of having counts at 20,000 being detected into the site of the tumor and not into anywhere else, we do see some in tail, and this is just due to the tail vein injection itself. So to test whether or not we're targeting this necrotic core, I excise the tumor out of the mouth, and then I cut it in half along the longest axis and open it up like a book so that the inner core, when I place it down on the image station, would face the camera. And then I just took an image, and we see that these high fluorescence intensities are heterogeneous and not homogeneous throughout the whole tumor. So with that, I then took a tunnel stain, which is just a common stain for cell death, and the cell death is indicated by the brown in this tumor slice. And this co-localizes very well with the fluorescence intensity that we see from the necrotic cells in this mammary xenograft. So I also used a rat model that contained a prostate tumor xenograft to validate what I saw with the mouse tumor model. So using our near-infrared PSS-794, I injected this via tail vein into the rat, and then 24 hours I took this X-ray overlay image, and we see that the tumor is clearly can be delineated from the rest of the body, and that these fluorescence intensities show a gradient, with the lowest amount being on the outside, and eventually getting higher and higher till we get to the core with the highest fluorescence intensities at this core of this tumor. So with this, I then excise the tumor and slice it in half, just like with the mammary xenograft, and place it on the image station. And this is the image that we received, and we can clearly see that these high fluorescence intensities are directly in the center of this tumor. So to really validate that we are targeting the necrotic cells, I did the histology. This is a bright field image of histology that we took from the core of the tumor, and we see that we see black irregular shaped cells, which represent necrotic cells, and this is easily contrasted to the living tissue in the tumor. We then took the same histograph and then took a fluorescence image on a microscope from it. And this is our Cy7 fluorescence from our PSS-794. And this co-localizes perfectly with the black irregular shaped cells inside the tumor. So this is extremely good validation that our probe is targeting the necrotic cells. Just like with the bacterial infection, we see significantly more accumulation of 794 in the tumor than with our control dye. I also did a H&E stain in the center of the tumors, and what we want to see here is that these black irregular shaped nuclei, and these indicate cell death occurring. We can also see that these cells appear to be irregularly shaped. I then took this H&E slice and looked under Cy7 fluorescence, and this is the fluorescence coming from our probe. And if we overlay these on top of each other, we see that there's perfect co-localization from the fluorescence of our probe onto these irregular shaped cells. So this once again shows that we can cells in tumors, allowing further detection in vivo. I've also been imaging tissue damage that is mediated by different cytotoxic agents. So in this case, we used an agent called a hydrophile, and this agent is an ionophore, so it's going to stick to the membranes of cells and depolarize them, which will lead to massive necrosis in tissues. So this is a montage of a typical study. We have 4 mice, and these mice were all injected with the hydrophile in this leg. And then 2 hours later, we injected our PSF 794, and then went through a longitudinal study like I've shown throughout this webinar. And just like what we've seen with the bacteria in the tumor, the probe diffuses and then clears away from the body and the site of the tissue damage, but then eventually it will just stick to the site of the tissue damage, allowing this to be easily detected in the body. And the beauty of the multispectral in vivo FX is that you can then take your fluorescence image and overlay it onto an X-ray, so you can get the exact anatomical location of the disease process that you're trying to target. I also wanted to determine if we could detect the severity of tissue damage. So using both a hydrophile, and then I also used ethanol in a series of mice. And I compared them to each other. So this is just 24 hours after our probe injection. And you can see the high fluorescence intensity from our probe coming in the hydrophile, and also our except these fluorescence intensities are not quite as high as they are with the hydrophile. And this can be shown in a 3D surface plot. We see that the fluorescence intensities are much higher, as depicted along the z-axis, than with the ethanol. And this, we then, using the target to non-target, showed that with the hydrophile, we have the highest target to non-target ratio, around 12 hours, which is a little bit over 6, and this is significantly higher than that of ethanol at 12 hours, which is around 3.5. So this is showing that with the higher target to non-target ratio, we're imaging the severity of the tissue damage, because with the hydrophile, there's more tissue damage occurring than with the ethanol. And the same can be said about the biodistribution graph. We have significantly higher probe accumulation with the hydrophile than with the ethanol. However, they're both quite high compared to other tissues in the body. So with that, I just want to say anionic membranes are good biomarkers for detecting and monitoring numerous disease states. And also, near-infrared fluorescence imaging allows for detection of your imaging agent in vivo without the background autofluorescence. And using the multimodal imaging with the Carestream in vivo multispectral effects, you can get a good anatomical map for delineating your optical signal in vivo. And that our zinc DPA fluorescent probes can localize to anionic membranes and bacteria, tumors, and tissue damage, both in vitro and in vivo. So with that, I would like to